thanks to Sophone for having me here, for inviting me along. I'd also like to give you a special thanks for promoting me to Professor. Unfortunately, my university doesn't know about this, so if you could pass that on, that's great. But essentially, don't believe everything you read in the programme. Okay, so as Tony just said, I'm going to talk to you about sodium channels. In particular, I'm going to talk to you a little bit about a couple of peptides that we've found in centipede venom that can modulate the function of the sodium channels. Okay, so centipedes, I don't know if many of you know much about these things, but they are actually venomous. They are venomous creatures. They have these first pair of legs here, which are modified into kind of fangs. We actually call them forciples because they're not mouth parts. And they use those forciples to inject quite potent venom. So if you do studies on live insects, for example, you can inject this venom and you very rapidly kill the insects after a bit of kind of increased activity. Okay, but very little's known about the venom. Only a few groups have worked on these, and there's probably 2 or 3 species where we know, a little bit of detail about this. If you get stung by one of these centipedes, okay, it causes excruciating burning pain. Okay, so that gives us some kind of leads as to what might be in this venom. Centipedes prey on large insects, some of these bigger ones here. This is a species called Scolopendra hardwickii. This is the species we've worked on. These will also prey on things like small mice, lizards, things like that, frogs. So they will prey on vertebrates as well. Okay, so that suggests to us they might have some interesting vertebrate toxins in there. And this particular species, they can reach sizes of around about 20 centimetres. Okay, so they're quite big animals. Actually, some people keep these as pets, believe it or not. Okay, they are quite dangerous. I wouldn't say they're ideal pets. They certainly don't fetch sticks back for you if you throw them. So, I probably wouldn't advise it. They can be quite dangerous. So in terms of collecting venom then, fortunately because these are big animals, they're quite generous when we milk them. So the way we do this is firstly very carefully, because we certainly don't want to suffer from any of these problems. So what we do is to knock these things out with carbon dioxide, push them into a tube. I don't know if you can see that in the background here. Push them into a tube that's continuously passing carbon dioxide through it. That keeps the centipede in a nice, peaceful state, at which point we apply a little electrical stimulation from this clamp behind the head, and then alongside the forciples there. And that gives us, as you can see, quite reasonable quantities of the venom. Okay, occasionally they come round during this process, which is a little bit awkward, but we've not had any problems yet, no injuries yet. Okay, so when we're thinking about targets that these centipede toxins might be aiming at, our first thought was, okay, sodium channels. One of the reasons we thought about that target is, of course, that if you look round through lots of other venomous animals and poisonous plants, things like that, then we find that a lot of those actually target sodium channels. Okay, so it's probably the most heavily targeted channel in nature from these natural toxins. And also there's a number of quite well characterised sites for action of these toxins. Okay, so a number of them here, 7 or 8 of these well characterised sites, depending whether you include these as one site, so the scorpion alpha toxin and delta conotoxin site. If you consider them as one or just overlapping, we have 7 or 8 really well characterised sites here. Okay, and of course, a couple well-known ones: the TTX site in the pore of the channel, binds to the pore loops, and of course scorpion alpha toxin, 2 toxins that were absolutely instrumental in our understanding of sodium channels, because of course they were used to purify sodium channel protein back in the 70s when we were just trying to get sequences of these things. Okay, so just bigging up the animal toxins a little bit there, just to tell you how useful they've been. Right, so our first little look was to just take some whole venom just to see if we actually see anything. And at the time we were working on this sodium channel from Drosophila. It's called the para-sodium channel. And we're working on that because we're interested in insecticides as well. And it's a target for what are known as the pyrethroid insecticides, also a target of a natural analogue of those, which are called pyrethrins. Okay, so we can express these in xenopusoocytes, and following some 2-electrode voltage clamp experiments, applying the venom, we find this massive gain of function, which seems to be not only an increased peak current, we get a much reduced extent of the fast inactivation, but also it seems we get this tail current as well, which was quite interesting for us because that's the way in which pyrethroids seem to work. Okay, so we were quite interested in those actions that we found. Okay, if we look at this current voltage relationship, again you can see that we get, apart from these actions we see here, we get this quite large negative shift in the V50 for activation. Okay, so it shifts it more than 20 millivolts, which is quite amazing. And of course this explains why when we inject whole venom into things like locusts, we get this kind of little burst of activity before they die. Okay, so a little bit of look to see what was in there. Are there any peptides, proteins in there? And the simple answer is that there seems to be a lot of them. But also what was quite interesting at this stage was that we, if you look at the top here, you see we repeated this experiment on several male and several female specimens. Okay, and what hopefully you can see is that there's a clear similarity within males or females, but there's quite a different profile between males and females. Okay, so that was quite interesting. I'm not going to speculate about the ecology behind that. That's not my area really. But we do get this clear sexual dimorphism. Also, if you sort of repeat those experiments with reduced or unreduced samples, it shows up some differences within males or females, and we kind of deduce from that that we've probably got peptides in there with a lot of disulfide bridging going on. Okay, so of course that's quite characteristic of these toxins, these peptide toxins. It's one feature that pulls many of them together. Okay, a little bit of mass spec then. So this is just looking at molecular weights below around about 10,000. Again, you can see that there's quite a lot going on, particularly in the males, in these kind of small to medium peptide ranges. Possibly more of the these smaller peptides in the males. Okay, but again, I'm not going to try and speculate about the ecology behind that. Okay, so we've got some activity there. We know that they modulate sodium channels. So the obvious next step was to do some fractionation of this. Okay, so we did some HPLC fractionation, which produced 96 fractions of each of male and female. Because, sadly, we were doing one cell at a time manual patch clamp, we didn't have any wonderful instruments to speed this up. We thought, to save this taking possibly 10 years, what we'd do is to kind of pull these fractions together. So for each of the male and the female specimens, we grouped them into 12 fractions each. So we pulled them into 8 separate fractions, each containing 12 of these original fractions. Okay, we tested them using now a patch clamp system, because obviously we're working with quite small amounts off these HPL systems, so we needed to scale down from the oocytes, of course. Okay, so we used patch clamp of TE671 cells, and these express NAV at 1.7. Okay, I'll show you this in the next slide, just to give you the proof of that. And then just using some simple traditional voltage protocols to look at the voltage sensitivity of activation and also of inactivation. Okay, so these are fairly standard practices for doing that. Okay, so yeah, just a little bit about this model system, which actually some of you might find incredibly useful if you're interested in NAV 1.7. But we've now shown that it almost exclusively expresses NAV 1.7 when it's in its undifferentiated state. Okay, so these are very easy cells to culture. They divide quite rapidly, and they're very easy to patch clamp. Okay, so really nice system for studying NAV 1.7. So just a little bit of evidence here. PCR shows that they're about 200-fold greater expression compared to what we expected, NAV 1.4. I say we expected that because these are actually derived from a muscle, skeletal muscle tumour. Okay, but of course we know that in tumour cells things can go a little bit astray. Western blot shows up NAV 1.7 as well, and of course if we look at several very selective toxins for NAV 1.7, particularly protoxin, that's really extremely selective for NAV 1.7, we get very low IC50 in the correct range there. So again, that might be a useful model for some of you that are working on NAV 1.7. Okay, so we started throwing these fractions or pooled fractions at these cells, and firstly looking at the activation. Okay, so voltage dependence of activation. We see here there's a couple of plots where we can see a significant shift to the left in the activation. Okay, so the channels are now activating at a more hyperpolarized potential. Okay, so that tells us we've got a gain of function, and this is looking a little bit like what we saw with the whole Venom. If we look at the female fractions against, sorry, the ones that I just showed you from male. If we look at the females, again we get, in this case, one fraction, which gives us this small leftward shift, but significant in the V50 for activation. If we have a look at inactivation, in the male, we've got a couple of fractions here which give this shift in the depolarizing direction. Okay, so we're getting quite reduced inactivations at these more positive membrane potentials. So these are the conditioning potentials, of course. Again, this tells us that we have overall gain of function in the sodium channels. Okay, and then if we look at the female venom against inactivation, again we see a couple of fractions where we get a little rightward shift in the curve, but also we can see here we get a small leftward shift, okay, in a couple of fractions there. Okay, so we've got here on the one hand gain in function and on the other hand loss of function. Okay, and you might think that's a bit strange. You know, why have you got two toxins perhaps in the same venom that have quite opposite effects? Well, actually, if you look at things like the cone snails are very well studied animal, produce a lot of peptide toxins. You see exactly the same thing there. You've got mu-conotoxins that inhibit the channel. You've got delta conotoxins that actually block the inactivation, so you get a gain of function. Okay, so we weren't too surprised by that. Also, if we look at the profile of these sodium currents, we can see that in some cases, well, just one case actually, this male fraction, we see quite a significantly increased rate of fast inactivation. Okay, so it's speeding up the activation, and again that's telling us that we've got an overall loss of function. Okay, so thanks to a good friend in University of Queensland, Ivan Dunheim, Irina, you probably know him very well. He took a couple of these fractions that we'd managed to separate and show some activity in, and very kindly produced some sequences of peptides in those fractions. So we got a little family of very similar peptides. Okay, so these four in particular, the first and the last three here were from this male 61 to 72 fraction, whereas this second sequence here was from an adjacent fraction, so maybe there's some carryover in that across those two fractions there. I guess features to look at is that they have this very well conserved cysteine pattern. Okay, so all of these peptides, although there seems to be a little family here and then a bit of a loner here, they have exactly the same cysteine pattern, and this is something that we see in other centipede toxins. Okay, so we picked on a couple of these to synthesize them and see if we can look at the activity of those. So we picked on the top one here and then this slightly different peptide here. Okay, so the first peptide, I've just called it SHTX1A. It will need a little bit of renaming once we're a little bit more familiar with its activity. But if we look at its effects on the activation, then we can see that we see this strong hyperpolarizing shift in the voltage dependence there. Okay, so that explains some of the activity we were seeing in the fractions and in the whole venom. Okay, and we get shifts at higher concentrations, about 100 nanomolar here, of around about -8 millivolts. The other peptide then, which I've called SHTX2, what we found with that one is that we saw no effect on the activation of the channel, but we did see this hyperpolarizing shift in the inactivation of the channel. Okay, so this is now kind of explaining the sort of negative effects that we had, that we saw on the sodium channel function. Okay, so these channels are becoming much more easy to push into the inactivated state. One thing's a little bit strange about this, although we see reasonable shifts in the inactivation curve here, we don't seem to see much concentration effect. So I don't know if we've got some incredibly potent toxin that's somehow saturated around this point, but we need to have a closer look at that. Okay, so finally a little bit of modelling then, some fairly preliminary modelling. Again, Ivan had already done some structural studies on this peptide and worked out the cysteine bridging patterns here, and we applied that to this toxin because it has exactly the same cysteine pattern. I guess the other thing to note with these peptides is that they've got an incredibly high density of negatively charged amino acids. Okay, there's a few positively charged ones in there, but there's this overriding kind of depth of negatively charged amino acids in both peptides. So that led us to believe that perhaps that's giving them the ability to interact with the positive charges on the S4 helices. Okay, so we know, if we go back to our model here, we know that the S4 helices are important for activation, but we also know that if we look at this S4 helix in the fourth domain of the sodium channel, this one is important for triggering inactivation as well. Okay, so I guess to cut a long story short, our kind of theory for how these toxins might be working is that the first one that gives us kind of eased activation is binding at this site here, close to where scorpion beta toxin binds, and by binding there it's kind of drawing the S4 helix out of its channel there. It's kind of encouraging activation, if you like. On the other hand, we think the same thing might be going on here with the SH2 toxin, but of course by drawing that S4 helix out, we are then encouraging the triggering of inactivation. Okay, so this is why we get enhanced activation here, but enhanced inactivation there. Okay, just briefly, we've done, again, with the help of Ivan, we've done a transcriptome, reveals about 415 peptide and protein sequences. 157 of those are neurotoxin-like. They fall into 17 families with good similarity and also similarity to other centipede toxins. And just looking at those sequences, we can predict that we may have some voltage-gated potassium channel and calcium channel inhibitors, a TRP1 agonist. Also based on, again, some whole Venom studies we did quite a long time back, we may have some nicotinic receptor inhibitors there. Okay, we also found beta pore-forming toxins, so it's punching holes in the membrane of the cells, and quite a number of enzymes. So a lot of these peptides look like they're stored as propeptides, so they need to be cleaved to release the mature peptide. Okay, and some of these enzymes might be playing that role. Okay, so just to wrap up, we've shown that we've got two NAV 1.7. I've put a question mark there because we've not done any selectivity studies yet. We don't know if they're selective for NAV 1.7 or not. Watch this space, hopefully. But certainly we've shown they target NAV 1.7. Positive modulator that may attract D2S4 out of its hiding place. And then an inhibitor which does the same in the fourth domain of the channel. Okay, so I guess it's just the tip of the iceberg. We've got lots more peptides that we can potentially look at. Just having pairs of hands and cash to do it, essentially. But we hope to move it forward soon. Just a few Acknowledgements then. Neville, he did most of the electrophysiology with the help of Amy Kerry. David Richards, he was our mad snake man who actually started keeping the centipedes for us. He was the only guy that was brave enough to actually look after them and handle them. David Tooth did the proteomics. Dong Hyun Kim, I've not actually talked about his work. There's lots of small molecules in there as well, neurotransmitters, amino acids, things like that, that we've not even started looking at those. Of course, Ivan Dunheim, very important guy for this project, helped us with the peptide sequencing, the transcriptome, and then Andy O'Reilly on the modelling. Okay, thank you.
Okay, time for some quick questions at the back here.
Thanks a lot, Ian. It's a very nice story. The VSD4 in the up and down position, it adopts really strikingly different electrostatic properties. So given your two toxins are, presumably it looks like one prefers the down state and the other prefers the up state. Have a look at the predicted distribution of the model of these two toxins, how the surface charge look like?
Not in great detail yet. So I just mentioned Andy O'Reilly, who kind of did this quick bit of modelling for us, a quick look-see, just to give us some predictions. But he's vowed to have a closer look at this. So yeah, it's a good point. It needs
many thanks.
Before we can put our hand on our heart and say this is what's going on.
It's quite interesting. Many thanks, Ian.
And of course, we need to try and qualify it with some probably mutational studies as well.
Okay, I think we've got time for one more question.
Yeah, hi Ian.
Oh, sorry.
I'm sitting right next to you. Nice talk. Did you ever try the toxins on sensory neurons?
We haven't yet, no.
Because the expectation is it would have a big effect on excitability.
Yeah. I mean, we perhaps plan to do that, particularly because there are some trip channel modulators there as well, we think. So,
yeah, I mean, you got a whole bunch of channels, right?
Yeah,
so you may be able to target.
It'd be a nice model to check a range of targets.
Really. Yeah.
I know another group in China, and I think in collaboration with Glenn King, they looked at DRGs, and this was in a different species, and found sodium channel inhibitors, potassium channel inhibitors, calcium channel inhibitors, but that's as far as they looked. And so, yeah, there's a little bit of information on other species. So.
Thanks.
Okay, I think we'll stop there.
Okay, thank
Thanks
you.
a lot, Ian. Thanks.